Welcome to week 8. This is the last week of Biology 216, section 2. Online, 8 weeks, you did it. This week we are going to talk about the reproductive system. The reproductive system basically has its own kind of function, whereas some of the other systems share functions, such as like we had some overabsorption and gas exchange and digestion and things. Well, reproduction is kind of its own function. So the reproductive system does its own thing. It is the only system out of all of them that we can actually live without medical intervention. So that's going to be the theme of this week. In lecture 1, I would like to review meiosis. This is going to be cell division that occurs in the sex cells. This should be a review for you from a previous biology class, but hopefully it's a much needed review. And so we're going to jump in here. So meiosis is a type of cell division for the formation of sex cells, gametes. So gametes are your sex cells, the sperm and the egg. So sperm for biological male, egg for biological female. These are formed in the gonads, which are the organs in which these are created, right, or mature. So in males, that would be the testes, and female, the ovaries. So sperm are formed in testes, oocytes are formed in ovaries. And sex cells differ from all the other cells of the body. All other cells of the body have 46 chromosomes each, 2 copies of each chromosome, but sperm and oocytes only have 23 chromosomes, so only 1 copy of each chromosome. And that's important because when a sperm and an egg join during fertilization to create a new embryo, 23 chromosomes here plus 23 chromosomes here equals that 46. And so all of our cells of our body, except for our sperm and our eggs, have 2 copies of each chromosome. So that's how it kind of differs. Meiosis is divided into meiosis 1 and meiosis 2. A little bit about terminology. This is where students get really confused. So we need to talk about what constitutes a chromosome, and what a chromatid is, and then what the centromere is. So when you look at this picture here, you see this little dot in the middle, that is a centromere. And we count number of chromosomes based on how many centromeres there are. And so here, we have 1 arm on each side of that centromere, but this is 1 chromosome. This is also 1 chromatid. A chromatid is kind of like a single arm here. Differing from here, so this is kind of that X-shaped structure that you're all probably familiar with. This is still 1 centromere, meaning this is also 1 chromosome, but this one has sister chromatids. So there's 1 arm on this side and 1 arm on this side. So 1 chromatid can be 1 chromosome, but a chromosome can also have 2 chromatids as well. It just kind of depends. But to make it easy, just count the number of centromeres, and that's how many chromosomes you have. Are there 1 arm or 2 arms? That's going to be your chromatid. So when we begin meiosis, the cell is diploid, which means it has 46 chromosomes, double, right? 2n, so double the amount. This is because the stem cells that sperm and egg arise from are diploid. Once they go through meiosis 1 and meiosis 2, they will reduce that number of chromosomes down to 23. So meiosis is sometimes called reduction division because we are reducing the number of chromosomes from 46 to 23 per cell. So when we begin, our cell is diploid, meaning we have 46 sister chromatids, okay? And those are all going to be replicated and then attached to their centromeres. So for example, I'm going to start drawing some pictures for you. Okay, so when we begin here, we have 46 of these little guys, okay? So there's 46 of these, each one is going to replicate. So if I were to draw out 46 of these and each one replicates, we're going to have a total of actually 92 chromatids, right? But these are also going to attach at the centromere. We'll get to that. So this is during interphase. So that's the phase of DNA replication. So all of these chromatids are going to replicate and make two chromatids. So then they're going to attach at the centromeres. So when those attach at the centromeres, now our result is that X shape with one centromere in the middle. So the result from this one chromosome made up of one chromatid, because we only have one arm here. We have one centromere, so this is still one chromosome, but we have two chromatids that are making that up right there. So essentially, we've kind of replicated the chromatid, if you want to think about it that way. All right, so these are homologous, so they're just direct copies of each other, which is important because then during meiosis, half of it's going to split, into one cell, and half of it's going to split into the other cell. And it'll be identical, right? So that happens during meiosis 1. Meiosis 2, we don't have replication again. That's the key between meiosis 1 and meiosis 2. During meiosis 2, we do not have replication again. So we're going to go over this in pictures and details. This is kind of an overview here. So in humans, we have 46 chromosomes, each with 2 chromatids, which is what I drew here. And so we're still going to have, oh my gosh, 46 of these guys, but 2 chromatids each. All right, so during the first meiotic division, what's going to happen here is that the chromosomes are going to pair up. So the male chromosome and the female chromosome, mom chromosome with dad chromosome, is going to pair up, and then they're going to split. During the second mitosis, and then you're still going to have a chromosome with 2 chromatids. Now, in the second meiotic division, now these are going to get split up into single chromatids per cell. So you can see here how the result after 2 rounds of meiosis, we went from 1 cell to 4 cells. Each of these cells has 1 chromatid per chromosome, and there's 23 chromosomes in each. Here we have chromosomes again, individual chromosomes, but they have 2 chromatids. They're going to be split. So let's go over this in detail. Okay, so we have this replication action happen. This is going to happen for each chromosome, and you have 23 chromosomes of your body. So every single one of those chromosomes is going to do this. Okay. Now, homologous chromosomes match up. What does that mean? Well, you had 23 of chromosomes coming from mom, and you had 23 chromosomes coming from dad. And these are going to pair up. So let's say this is like for, let's say chromosome, I don't know. Chromosome 12, for example. Well, it doesn't matter. They're all going to do the same thing. So this is chromosome 12 for mom, and this is chromosome 12 from dad, out of 23. And these are going to match up. So they're going to come together very close in proximity to each other. And what they're going to do is begin to exchange genetic material. It's called crossing over. So part of this chromosome might come over here and swap with this. This part might swap over here with this. That's called crossing over. And the point of that is to increase our genetic diversity. So now, instead of all of this genetic material being maternal and all of this genetic material being paternal, what you're going to have instead is chromosomes with both parents' genetic material on each. Okay. So now this chromosome has some genetic material from mom and dad. This chromosome has genetic material from mom and dad. That's why you get some traits from your mom, some traits from your dad, and all your siblings are going to get different traits, right? Because it just depends on which of these chromosomes ends up in the embryo. So that's called crossing over. That's going to happen in prophase 1. And now we're ready to go through cell division. So what these are going to do are line up in the middle of the cell. Let's see here. So here's our cell. We have things called spindle fibers. That's going to end up pulling these chromosomes apart. And we're going to have our chromosomes line up like so. All 23 of them will line up. I'm just going to draw one for now for simplicity. And what's going to happen is these are going to go to opposite sides of the cell. And that's going to happen. So this is metaphase 1 when they all line up in the middle of the cell here, and there'll be more here, right? Then these are all going to be pulled this direction. These are all going to be pulled this direction. And that is for anaphase 1. So now we have had chromosomes go to each side of the cell. Here. Then in telophase 1, cytokinesis means splitting of the cytoplasm. So that's kind of the official 2-cell result. Then what's going to happen here is that we have 2 cells, each with the chromosomes that got pulled to the side of the cell. Okay, so now we have 2 cells. They all have 2 chromatids, but we have 23 chromosomes per cell. Up here, we had 46 chromosomes in this cell because we had 2 copies of each in each cell. Down here, we just have 1 copy of each in each cell. So that's only 23 chromosomes. Okay, that's our first reduction. So just to kind of recap, we had chromosome from mom joining up with chromosome from dad. Okay, we had crossing over occur. Well, first what happened is we have this, because this is what you start with, right? Each of these chromatids have to be replicated into this X shape. Then the X's from mom join up with the X's from dad. Then we have that crossing over occurring, and they all line up in the middle of the cell here. Then we have them splitting to each side of the cell. And now we have 1 copy of each type of chromosome in each cell. So this is chromosome 12 again. So there's a copy of chromosome 12 over here. There's a copy of chromosome 12 over here. Maybe you call this chromosome 2. Maybe you call this chromosome, I don't know, 10. It doesn't really matter, right? You're going to have 1 copy of each chromosome in each. That's 23. Now, we're going to go into meiosis 2. There's no additional DNA replication here. So this isn't going to happen. Why? Because we already are in our X format. Okay, we don't need to go into another X format. If we were to duplicate this again, we're going to have extra chromosomes, and some genetic abnormalities actually occur from that, which is kind of interesting. So what's going to happen next is we're going to go into meiosis 2. The goal is to get back down to this scenario, okay? Because that's how we started, right? We replicated this into 2, and then it went into an X shape, right? We need to get back to this. This is what can be passed on. All right, so prophase 2, what's going to happen? Pretty much all that's going to happen is those spindle fibers are going to form again, since we don't have any matching up, because there's not a second copy to match up with, right? Let's see here. So essentially these are going to kind of just be hanging out, and the spindle fibers are going to form. That's prophase 1. Metaphase 1, they're going to line up in the center of the equator here. So I'm not going to draw that again, but essentially they would line up in the center. Anaphase 1, now what's going to happen, draw this bigger, is these are going to separate. So now we're going to have let's see. Something like that. And this one would look like this. This one would look like this. This would have a little bit there, and this one would have, let's see, some here. All I've done now in anaphase is split these right down the centromere. And look what we have now, single arm genetic material. That's our goal, right? So what's going to happen here then is that, and this is happening for two different cells, I should say. So then let's see, hold up just a second here. Oh yeah, here. So essentially that's happening in two different cells. So this should actually be one cell. This should be one cell. Let me back up here real quick. So sorry about that. So essentially, let's see, meiosis 2. Let's back up. Let's draw what went into meiosis 2. What went into meiosis 2 is we had a chromosome that looked like this. See, this is confusing. A chromosome that looked like this. Now, each of these are going to undergo anaphase. Okay. Then this is going to be split down the middle. This is going to be split down the middle. And you'll have this, 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 and this in your 4 cells that come out of telophase 2. Sorry, I kind of confused you all. But essentially, this is what's going to result from telophase 2. You're going to have 4 different daughter cells. Each with 23 chromosomes. How many chromatids do they have? They each just have 1. And these are going to be our sex cells. So each of these has 23 chromosomes. When they join up with the sperm chromosomes, right, they're each going to replicate and join and match up with the sperm chromosomes. And we're going to do the whole thing all over again when you make a new embryo. All right. So this is just an example of crossing over. So this is basically an example of eye color. So for example, maybe you have a different eye color than your siblings have. So here's your chromosomes from mom. Here's your chromosomes from dad. And let's say that, oh, we have blood type here too, right? You have blood type gene here on this arm, on these arms. You have hair color here, eye color here. And your mom passed on her genes for all of those things, and your dad passed on his genes for all of those things. What's going to happen during crossing over is maybe dad's blood type gene swaps with mom's blood type gene. All right, so then what's going to happen then out of meiosis 2 is going to be, you have the potential, okay, to have mom's full blood type and hair color and eye color. You might have dad's blood type, mom's eye and hair color. You might have mom's blood type and dad's eye and hair color, or you might have everything like your dad. And so whichever one of these prevails to fertilization, that determines what your genes are going to be. So that's why maybe you got everything from mom, but your sibling got something from dad, and so on and so forth. It just kind of depends on which one of those 4 cells will end up passing along their genes. Point of crossing over is to recombinate. If we just had the opportunity to pass on mom's genes or just dad's genes, the other parent's genes will never get passed on. And so that doesn't create genetic diversity. And that happens a lot in like incest populations. So why do we need to do 2 rounds of cell division? Well, first of all, if you pass along 46 chromosomes, your offspring will have double the amount of chromosomes, right? They'll have 92 chromosomes per per child, and that's not good. Each human has 46 chromosomes. That's the way it is. But more importantly, it improves the gene pool, right? So we don't want just our genes from mom to be passed on and just our genes from dad to be passed on. We want a mixture of them. That provides for 8 million combinations of 23 chromosomes, can combine with 8 million combinations of 23 chromosomes, which can generate 70 trillion genetically diverse individuals. That is insane, right? That's a huge gene pool. And so that crossing over just adds more variability to that. And what we're going to talk about here with how sperm are formed and how eggs are formed is that they do so on a different timeline. So they're both going to go undergo those 2 rounds of meiosis, but male sperm at puberty continues through both meiosis 1 and meiosis 2. So those sperm are mature at puberty and capable of fertilization. But female oocytes actually start in utero when you were in utero and you were developing inside your mother. And so that's when prophase 1 starts. Prophase 1, meiosis 1. And then at puberty, that's going to complete, and meiosis 2 actually doesn't ever complete until fertilization, if fertilization occurs. So it's a much longer drawn out process for egg development rather than sperm development. All right, so that is just a quick rundown on meiosis. Hopefully that was kind of a review. I just want you to have a background on why gametes have 23 chromosomes and how that occurs. Remember, if you get confused, this can be very confusing, but just remember that you have genes from, or copies from mom and copies from dad. And then hopefully that makes it a little bit easier, but please let me know if you have any questions.